We are going to discuss various approaches to drug design, and we are going to discuss this in parts. This is part 1. We are going to deal with the historical to the latest approaches. Drug discovery is very important, and particularly you can find the whole world is trying to find new drugs. Forget the latest one is the COVID. We are going to discuss what is drug design, what is a drug, what are various approaches. We are going to talk about these various approaches in details, 6, 7 traditional and some little bit rational approaches, and then we are going to discuss in detail the Hans approaches of S.A.R and their parameters and the methodology, 3D S.A.R and molecular modeling, pharmacophore modeling, docking, and high throughput screening. What is a drug? Can we say a drug is any synthetic or natural product which is substance, any chemical? Sugar is also chemical. Can you call it as a drug? Or drug which alters or affects any microorganism? So even physically, if an axe can break a limb, can it be called as a drug? Or drug which brings about some enhancement in functionality, which stimulates your cup of coffee, can you call it as a drug? In a very specific sense, we will differ in that. We need a more specific, definition, and particular points like potential use and for diagnosis and treatment of diseases and disorders, and should have specific action, not a general action. You don't have a drug in the market which is used for 5-6 diseases. So the US Foods, Drugs and Cosmetic Act has defined drug as a substance used in the diagnosis, treatment and prevention of disease or as a component of medication. This is most simple and valid definition of a drug substance. Why do you need drugs every now and then? Today also we don't have drug for COVID. Few years back we did not have drug for SARS, and we did not have for swine flu or even for AIDS. Our ancestors did not talk about cancer. And this type of diseases keep on cropping. New diseases like AIDS, Alzheimer, obesity has become a big, diabetes has become a big disorder. So you need to find better and better effective drugs. Dementia, cancer. There are some people who are neurotic, they need antidepressants, which give a lot of side effects. Sometimes downstream health cost of therapy, and cost of to individual and countries to sustain industrial. These are all unmet demands, and so you need drugs. What is drug designing? These are various approaches to develop a drug or find out a drug. These are various approaches like from age-old days, random synthesis and screening of products and natural products. Then we came up with till last 50-60 years back, people used to do isosteric replacements, and they used to do S.A.R. If there is a benzene ring, replace it with thiophene and find out, or change some substituent and find out. Then came analog based drug design, more rational, with Sir Corwin Hansch from 60 onwards, and people started depending on the physicochemical properties and designing drugs. More rational approaches came of structure based drug design, and there are today many like genomics and other things. These are one and same like the historical approaches. The Chinese emperor was suffering from chill and fever and tremors, and so he gave him cinchona bark powder, and this is how quinine came in existence as an anti-malarial. Some came by chance and serendipity like penicillins. S.A.Rs were developed from natural products, and you got propranolol and cimetidine, etc. Some came by systematically studying the disease process, and tamoxifen came for breast cancer, L-DOPA came from Parkinson's disease. Biological and physiological pathways, biochemical were studied, and you got ACE inhibitors when you studied the renin-angiotensin and system, and then angiotensin converting enzyme inhibitors, and same thing. Then receptor came, and this ACE was one of the earlier enzymes as a receptor site. People came with n-mass synthesis and screening in fractions of hours, automated. That also yielded some real good leads. And today we are talking about genomics, recombinant DNA, and genetic engineering products. Let us visit the traditional approaches, which are were based mostly on serendipity and intuition, and they were not rational, and they never led to de novo drug design or breakthrough. We had homolog approach, like you go on making changes at this uh, you have repeated changes in the methylene group. You will come up with homologs and synthesize and test them. You had some natural products like morphine. Why you need 5 rings? So break one and check. And people found out pentazocine, meprobamate, which are simpler than morphine. Some people went for making additions to the existing drug molecule to find whether there is a change, whether there is that at that time receptor concept was also known, so there is any modification in binding with the receptors. This is how morphine antagonist nalorphine was discovered, and people found out isosteric replacements and bioisosteric replacements, and PABA. From PABA came sulfonamides. This is an example of carboxylic acid. The isosteric groups are many. Sulfonamide is one of them. Tetrazole is another of them. And those isosteric groups, which also give similar biological properties, were called as bioisosteric groups. Came simple structure activity relationships in picture. People depended on them for few centuries. And if you make this variation, you need this group to be here. Like for example, in pyrimethamine, you need a 5-chloro substituent on the phenyl ring, of para-chloro substituent on the 5 position phenyl ring, and you need both the amino groups, 2,4-diamino groups to be free, and at 6th position, nothing less or more than ethyl group, otherwise activity goes down, and these were all called as S.A.Rs. And then people started finding leads to improve the biological activity, the potency, the efficacy, to reduce the side effects, to improve on the ADME characters, and many of these features like molecular structure, its solubility in the biological fluids, its complementarity with the receptor, its favorable interaction with the receptor, structural changes when it binds with the receptor, and the observed elicited biological response. Um, there are various approaches which we are going to discuss now. Is the Hansch analysis in details, the free Wilson and de novo method, the Topliss operational scheme, the crate plot, the cluster analysis, the Fibonacci search method, the simplex method. We will see Hansch in more detail afterwards in the second part, but we will see these other approaches, okay, one by one in 2 to 3 slides maybe. So free Wilson is basically more a statistical model. You can use a variety of substituents. Don't bother. You are not bothered about the physicochemical properties. There are few conditions, 5 conditions which are to be followed, principles which are listed in the next slide. And as long as you follow that, it becomes tedious sometimes. You have to do a lot of synthesis and other things. And there is statistics also involved. Suppose this is a tetracyclic structure, then every time you should stick to this. All analogs should have this skeleton first thing. If these are 2 positions of substitution, R1 and R2, then all analogs should stick to this. If anything is having another substituent, it will be an outlier. Cannot be taken in the study. Each substitution is believed to make some contribution, positive or negative. And the biological activity is basically coming from the basic skeleton, which is designated as mu, and the substituents at different positions, and the contribution of the substituents their presence and absence of the substituents at these positions, and their positive and negative contribution. So this is biological activity contribution, the presence and absence, and this is the basic skeleton. And the most important is the fifth, the principle of symmetry or the restriction equation. Supposingly I have 5 substituents here, then if one is contributing say 20% and another is contributing 30%, then maybe the others are contributing -20, -20, -10, so the sum total should be 0. It is to be believed that the total contribution of different substituents at a position should be 0. So it is believed that, or it is assumed that some of the substituents have positive and some of them have negative contribution. Here we have taken a free Wilson matrix, we have taken a specific tetracyclic compound, and we have X, Y and R positions, and we have made a first preliminary table or matrix number 1 with compound number 10 compounds, their biological activities and their substituents have been listed here. So for at R position we have 2 substituents, H5, 6 we have H and 4 we have CH3. At X position we have 3 substituents, nitro, chloro and bromo, and at Y position we have nitro, amino and acetylamino. Let us expand this more systematically, and at R position we have 2, at X we have 3 and at Y we have 3. Wherever whatever is present we have put 1 1 1 1 1, sum total we have done. Now for R position, like for R position, we come out with 6H and 4CH3. Okay, so we have 6H plus 4CH3. There, as per the symmetry principle, contribution is 0. So we express them in terms of each other. So 1 CH3 is equal to minus 1.5H. On similar lines, we can express chloro in terms of nitro and bromo. 1 chloro is 2 nitro, minus 2 nitro, minus 2 bromo. And acetylamino can be expressed in terms of amino and nitro. So 1.5 nitro minus 1.5 nitro minus 2.5 amino is acetylamino. Now exactly this matrix is further expanded, and what we do is we are expressing wherever, for example, wherever is H we have put H, but we are removing CH3, we are removing chloro, we are removing amino, and these things are expressed in terms of this is expressed in terms of this 2, this is expressed in this 2. So instead of 1 2 3 4 5 6 7 columns, now we will have lesser columns. Okay, so we will have here 1 column because CH3 is expressed in its term. We will have here 2 columns and we will have here 2 columns. So we will have 5 columns for the substituents. And so here we have expressed wherever H was there, we have put 1 1 1. Other CH3 was expressed in terms of H minus 1.5H. Here in nitro bromo, wherever chloro came, we expressed it minus 2 2, because we see here chloro is minus 2, combination of these two. Okay, so like that we filled up this and we took the log of biological activity. Now this we did it in a multiple linear regression analysis using a computer software, and we can come up with a mathematical model. We have another approach called as Topliss scheme, which is very helpful in finding out in one by one stepwise selection of permutation combinations of mainly 2 properties or 3 properties, that is you can say lipophilic, steric and electronic properties. Accordingly you choose the substituent and observe what activity you get, and accordingly then you reduce one of these activity which is not favorable and proceed further. How? I will show you. There are 2 types of substituents, aromatic and aliphatic. This was particularly devised by John J. Topliss and P.C. Martin. And basically They relied more on the hydrophobicity and electronic factors than the steric factors. And how it is done, I will show you. They found a root which is finally designated as a Topliss tree. They started with an unsubstituted compound on the benzene ring at the para position. They made a first change 4-chloro. So there are three possibilities. You got more activity, you got equal activity, you got less activity. If you got more activity, that means chloro is helping. So in this step, what they have done is they have increased both the lipophilicity as well as the electronic parameter, sigma and pi both. So they added one more chloro. If they got still more activity, they further increase the electron withdrawing power and the bulk. If they got equal activity, then there's a rethought. Maybe we need the bulk, but we don't need the electron withdrawing power. Maybe electron donating may be better, so they replaced it with CH3. If they ended up with increase, then they were true. Their thinking was true. They further went with third butyl. If there was not significant change, so maybe the position of chloro had to do something, so they just changed the position of this chloro. If activity was less, also position of chloro. And again, if they got activity good, they three positions they stuck and they made more changes. They increased the number of fluoroatoms and the bulk. And by chance, if they landed with less activity, then they made a polar substitution. Nothing much happened, then they changed again to 3-chloro, but if they found activity increase, they made it more polar and more polar. And this is how, wherever they landed with the best activity, they selected the substituent. Here same thing has been explained for aromatic, how they went on increasing the pi and sigma, but increasing pi and decreasing sigma, and both decreasing like that. Same thing they have given for aliphatic also. You start with methyl and make a quantum change of isopropyl, but if nothing happens, come down to ethyl, more, take it bulkier group, but less, make it more polar, and so on and so forth. Okay, like that they went on slowly. Another methodology was Craig's plot, named after the scientist Craig. And you want to select a very good series, so very good selection of substituents is important to make it more generalized, your study to be more generalized, your findings to be more accurate. And so under such cases, what happens is you need to take care of both parameters, sigma and pi. So at four corners you will have positive sigma, positive pi, negative sigma, negative pi, one of them alternatively positive, negative, negative, positive. That is what is with alkyl. You will have positive pi but negative sigma. Halogens you will have, okay, your lipophilicity and electron withdrawing power both will increase, but with hydroxyl your both will decrease. So these are the possibilities: plus, minus, plus, plus, minus, 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 plus. Okay, so all four corners they have drawn a very nice graph and place the substituents here. So if you pick up 2, 2, 2, 2 from each and make a series of 8 compounds, or 3, 3, 3, 3 and make a 12 compounds, you will get more accurate series, and you can plan a synthesis and screening, and your results will tell you more, throw more light that which parameter is really bothering the biological activity. You will get more generalized and more covering evidence. On similar lines, Ewan Connelly Martin once again teamed up with Sir Corwin Hansch, and she found out subclusters for aromatic and aliphatic substituents. She found out groups of aromatic and aliphatic substituents, subdivided them into 10 groups. An idea was when you plan a series, you pick up one from each. So your series will be covering all corners as you saw in the Greek plot, and your study will be more rational and more accurate. So she has actually suggested that we pick up for aromatic aliphatic. Next preceding slides we will find both the clusters, and each cluster one you select and synthesize and test. If you get some results, next time you select the next. This is the aromatic cluster, and this is the aliphatic cluster, and that's all. Simplex method is again by Darvas. It also searches the more active compounds in different directions. Whichever direction you get less activity, it moves away from that direction. And how it does it, it draws a triangle, and then further it goes on reflecting or contracting the triangle. I will show you here. Supposingly you have three compounds A, B and C on a energy surface diagram of log P and pK. Okay, and you check synthesize their values. You can see that log P is different for all three, and pK is also different. So three different sets you have got of the lipophilic and electronic properties. You synthesize, then you find out. Supposingly if you find A is the least active analog, then B and C compared to. So you draw a line through B and C as if it is a mirror at the same distance, and locate one point with new pK and log P values, and find out a substituent and synthesize that compound. Suppose if you get it more active, you proceed further. So you are from here, you are going here. So you are expanding your this thing through the reflection. Okay. But suppose your activity was lesser than this, you return back. So you are contracting. Okay. Same thing you can do. Instead of A, you can go for B. B was the least active. Then you could have gone beyond this A and C. You to B double dash, or you draw a new triangle with this and you go beyond that. Or suppose if C was the least active, then you would have gone like that to C dash, and like that. So you do continuously reflection and contraction and find out the space. Okay. So Fibonacci search I will not deal, because a separate YouTube video I have posted on the YouTube. So I think this was just an introduction. We covered various techniques, and 6-7 techniques from traditional to the till the QSAR. QSAR we are going to deal with separately. Thank you.